Oh, so
Maybe we should
in 2
start?
minutes it'll start. Yeah.
I just started it now, though. Is it
Counting
live?
down.
Oh.
It's gonna count down.
So there's a start time.
Yes. Apparently YouTube is counting down. You're good to go.
I know, I started early.
Nice.
This is intense. Is there?
10-second delay. Basically, start swearing or
I'm not fixing anything. I'm done.
wardrobe malfunctions or something. Half the time I was.
Holy crap, you Hey, there's a whole bunch of coffee, by the way, if you need to get water. I'll get water. I'm gonna stand here so you're done with this.
Bill will probably be proud of it because before I was very easy. I don't have to. I think not mine.
I didn't print anything out, Jay, I'm sorry.
Okay. No, we okay, I think we'll get started. Thanks for showing up. Welcome. It's my pleasure to introduce Mario Muscarella. Perhaps many of you know him, but I want to give you a little bit of background, talk a little bit more about how he's been kind of an integral member of the lab here at IU, but also during this transition from when we moved here a couple of years ago as a lab. So in 2008, he got his bachelor's degree in biology from Armstrong State University in Georgia. And after that, he spent a couple years working at the University of Georgia's Marine Biological Station, where he got a lot of expertise and basically sampling marine systems, learning how to do basic microbiology. Actually, things I think that have kind of helped him take parts of his thesis and move through them very, very smoothly. And then in 2010, he came to Michigan State University, where he was admitted into the Department of Microbiology and Molecular Genetics on some sort of distinguished student fellowship so to encourage good students like him to come to MSU. And so in reflecting on this, I realized he was only there for 2 years out of the time that he had been a student, and we uprooted our lab and we moved here. And I think, I personally, I'm not sure about other students, people in my lab, but I never look back. It's been a great move. But I remember talking with Jeff Palmer. I come back on a second visit, and you're trying to figure out, like, are you going to move or not? There's a lot going on in terms of like the lab physically moving, your personal life, all these things. And Jeff said to me, it's like, there's no way you should do this unless you get some people in your lab that are going to help you and come with you. And so that was critical. And there's Megan was one of those people. Mario was the other person. So I just kind of want to highlight some of the things that triggered my memory when I was coming up with this today. One is that we had just had a baby, and so I had to move by myself because my wife's like, I'm not going down there. You can do the move on your own. So we got dropped off at Boulders Deer Park, which is an awful apartment rental in case you want to know. And they dropped our pod there, and the only person who was there at that time to help me physically move things into my new apartment was Mario. So that was really nice. And then whenever I put a student on an RA in my laboratory, they're required to do something that's not related necessarily to their dissertation research. And so part of getting the lab set up was that I wanted to get these different systems So Megan had a system, Mario had a system, and you can make this really complicated in how you want to develop a system. But I took the opposite approach with Mario. I said, at the end of the semester, all I want to do is see a picture of a gel with a 16S ribosomal RNA gene product. He's like, oh, this is easy. I know how to do this. This is no problem. I said, okay, good, great, go for it. And then he's like, I'm like, here's the account number. He's like, well, what do you mean? Where's the equipment? Where's the pipetters? Where's the reagents? Where's the gel box? All this stuff. And so as he moves on in his career, if he ever has an opportunity where he has to set up his own lab, he should be in good shape. This is my mentoring plan.
You're
right? This is how I train my students. I think I want to comment on a couple areas where I've seen Mario grow a lot as an individual and as a scientist over the past 6 years or so. One was we went to University of Michigan this is right about the time, 2010, where a lot of people were doing a lot of community sequencing of bacterial communities, which you're going to hear a little bit about in Mario's talk today. And people weren't really doing this at the same degree, at the same throughput, the same speed that James Ford, for example, can get us samples back in a day. It would sometimes take months to get these things going. Then, of course, you're opening up these really large data files. And a lot of the pipelines that we now take for granted just a few years ago weren't really available. So I said, all right, a couple of people in my lab have gone to University of Michigan where people who will train us how to do this. And so Mario and I happened to go there, and it was at the Detroit International Airport. So we went into this conference in this really bad hotel where we learned how to do this stuff. And at that time, I actually had a little bit more experience, I think, than Mario did. But after that 3-day workshop, it was all over. Like, he's just picked up a lot of this stuff and really, not only has he got it working for his own research, other people in our lab are using this, and just the skills that he's picked up from that have really just kind of grown exponentially. So his computer skills have really taken off, and in the process of that, he's helped with teaching courses both at Michigan State and also the quantitative biodiversity course that we developed here a couple years ago. So he's really taken off, and I think his new postdoc experience is just going to build off of that. He's done also a lot of field work. You're going to hear a little bit about that in the Upper Peninsula of Michigan, which I think from here is probably a 12 to 14-hour drive. The first year, Megan and Mario went up together, but then after that, he was renting cars and just driving up there and hanging out by himself, working under probably not the most ideal conditions for collecting the data. So there's a lot of challenges and a lot of improvisation that he needed to employ to kind of get the data that he's going to talk about today. He's really good also in the laboratory, and he's in the process has mentored a lot of undergraduates and high school students in STEM programs and such. So that's been another thing that I've seen him take on as a senior grad student here at IU. I think he's also been a part of something that's been a little bit more lab-wide shift in the culture, where people are a lot more open in sharing with their data, their code, how they're thinking about things. We've used certain tools that he's helped develop that I think really, I've noticed, and maybe not everyone else, probably Megan and Mario, but it's really changed a lot, probably coincides with our moving here. So I think the culture has changed, and people like Venus who are coming into the lab probably aren't going to have the ability to reflect on that, but I think Mario's contributed a lot to that. I'm going to get going. Accomplishments, he's got a number of grants, one from the Huron Mountain Foundation up in UP, Indiana Academy of Science. He was awarded a Dissertation Improvement Grant last year or the year before that. He's given lots of talks at national meetings, the Ecological Society of America, regional meetings like NEEC, and he was a grad student liaison for the Ecological Society of America's Microbial Ecology Society. He's got 6 peer-reviewed publications. 2 of these are first authored, and they're part of his thesis, so that's a great thing to see happen. So what's next? Everyone wants to know at this stage, like, what are you going to be doing next? Very shortly, I'm not sure which one of these things is going first, he was invited to go to Barcelona, where he's going to be participating in the Ramon Margalov Summer Colloquium on Microbes in a Changing World. Soon thereafter, he was recently invited to another workshop at the Santa Fe Institute on disturbance in the human microbiome, where he's going to be giving a talk there. And then in mid-August, he's going to be starting a postdoc in James O'Dwyer's lab. Some of you may remember he came here a couple years ago to give a seminar. I must have been impressed with Mario at that time. Went and gave a seminar, the departmental seminar there. So he's going to be switching gears a little bit and perhaps putting his muddy boots aside for a little while, perhaps not permanently, and learn a little bit more about theoretical approaches to the phylogenetic community ecology of microbial systems. So with that, we're going to learn a little bit more about microbes and resources. And again, thanks for coming.
Thank you, Jay. Jay failed to mention that I had to apply to work with him 3 times. And I rented a lot of U-Hauls while being a grad student, but we'll talk about that over beers. Okay, so perfect. I am really into open science, so we're live streaming this. I hope everyone's okay with that. That's the first disclaimer. I'm also really into acknowledging people before I talk about my research. I have a great committee: Spencer, Jake, and Rich, and Jay, of course. Thank you, Jay, for the kind words. Brent, who's our lab manager, has been instrumental in making most of my research work. He's helped me troubleshoot things. I had a really talented undergrad, Shimon, who helped pretty much gather most of the data for an entire chapter in my dissertation. I have fabulous lab members like Megan and a former lab member, Callie, who are co-authors of papers. These people are instrumental when you move universities. You don't realize how important the secretary, the department chair, the former department chair, and at that time, the acting grad director are, and moving all your coursework between one university to another so you don't have to restart your entire PhD. That was pretty stressful. Most of my research was also influenced by my committee at Michigan State, which consists of Tom Schmidt, Jim Teege, and Steve Hamilton. And then I have a long list of collaborators, co-authors, peer reviews, people who help me learn to write. Ariane Peralta just drove here from North Carolina, which is pretty amazing. There's great funding agencies, and we can talk about that later. Oh yeah, so I need to thank my funding sources. The Huron Mountain Wildlife Foundation is this private research foundation that owns property in the Upper Peninsula of Michigan. So if you have like the glove of Michigan, there's this other part you forget about, and they're way up here. They've graciously given me a lot of money. National Science Foundation awarded me a doctoral dissertation improvement grant, which funded part of my research and improved a lot of it, actually. I got funding from the Indiana Academy of Sciences and fellowships from Michigan State and Indiana University. So if I left you off, I'm sorry. Let's have a beer instead. And let's get going here. And so I want to start here. This isn't space. This is a stained image of bacterial cells, or their DNA stained in surface water on the globe. You go anywhere on the globe today and you get a mill of seawater or lake water or river water, you'll probably find about 10 to the 6 cells per mill. And those 10 to the 6 cells represent thousands of different species that are sort of coexisting. And recently there's been some interest. There was a paper came out earlier this week bringing up Hutchinson's paradox of plankton. How can you get so many individual organisms to coexist at once? And I would argue that microbes are maybe perhaps the best example of this, where you can maintain thousands of species in surface waters across the entire globe. And they're sort of doing something, right? We have a traditional view of biogeochemistry that they might be bags of enzymes that are processing nutrients in the environment and contributing to the cycling of carbon, nitrogen, breaking down phosphorus compounds, moving energy into the food web, and dissipating CO2 in the atmosphere as they respire. But I'm really interested in what is enabling them to do that. What controls who's there, who's active, and what they're doing. And that is a little bit about what I want to talk to you today. So we know microbial communities are very diverse. We can go pretty much anywhere on the planet. We can take a sample. We can sequence it. This is great. The sequencing revolution has changed how we do microbial ecology. The Earth Microbiome Project has shown this to us immensely. We just sequence anything. And we can use marker genes like 16S to identify who might be there and their relative abundance, or maybe even their relative activity. See who's active in the habitat and who's not active in that habitat. I'm not going to bore you guys today with all the details about how I sequence things. It's changed a lot in the 6 years I've been a grad student. It's gone from being able to produce hundreds of thousands to now millions of sequences within the night. So I'm just going to tell you that throughout my talk, when I refer to microbial communities, I refer to where I've gone in and I've sequenced the 16S ribosomal RNA gene, and I've used that as a marker gene for the diversity of organisms in that sample. All right, so we know across habitats, microbial communities differ. We also know that resources differ between those habitats. These are the chemicals that maybe those microbes are processing in the carbon cycle, for instance. Or maybe they're transforming nitrogen into new forms and there's denitrification, all these interesting biogeochemical processes happening. But to do any of these organisms have to have fundamental resources to act upon. If they're heterotrophic organisms, they rely on those resources for energy as well as the molecules to build new biomass. So resources are fundamental to who's able to survive in a habitat and might distinguish what organisms are in one habitat versus another. But resources aren't just carbon, nitrogen, phosphorus. They're actually pretty diverse. You might have different concentrations of the same resource. So here I'm showing dissolved organic carbon as a gradient. You might have more dissolved organic carbon or less dissolved organic carbon. But you also might have a diversity of different chemicals. And this is important because these organisms are osmotrophs. They take in dissolved chemicals from the environment, and they need to break them down to get that energy and to form new biomass. So the form of the element or the form of the resource available to them might depict who's able to do what in a given habitat. Now, I think this goes underappreciated often, and we're now understanding that there's a diversity of chemicals in any given environment. We can use high-resolution mass spectrometry to go into any environment and let's say look at the dissolved organic matter. And now we've gone from measuring the concentration of dissolved organic carbon or the concentration of total phosphorus or total nitrogen, and now we know that there's a diversity of chemicals that possibly these organisms are using and might distinguish between one organism and another. One organism may not be able to use a certain chemical. This isn't novel to microbes. Resources are diverse across all, the entire tree of life. You can go from organisms that use nuts, organisms that might specialize on pollen. You can even have different phytoplankton that use specialized wavelengths of light in order to partition resources. This is a fundamental mechanism of how organisms differentiate themselves. There's this ecological specialization on resources. I'm going to use this resource that my neighbor is not using. Now we can both maybe coexist. Now this is just one of the many coexistence mechanisms there are for communities and one of the possible processes by which communities assemble. But this is fundamentally what I'm interested in my dissertation. Now today I want to talk about two cases of resources, and I want to talk about how resources between two habitats can differ and how this can affect microbial communities. For instance, I can have a habitat that has all blue squares, and I can have another one that has all green triangles, and maybe there's some organisms that like blue squares and green triangles. I'm also going to talk today about how heterogeneity, this variation in resource type, within a habitat can affect microbial communities. So given this habitat here, I might have an equal mixture of squares, triangles, and hexagons, and maybe there's different organisms that like each of those resources. Alternatively, the little triangle resource might be really rare, might not be available to most consumers. So there might be differences in the relative abundance of different resources in a given habitat. And last, you might have a habitat where one resource is just not there. So you might have a variation in the types of resources that are given there and also their relative abundances. I'm going to start by talking about this between habitats. So habitats that differ in the resources that are present. And say in a situation where you might have three environments and they might have different resources in them. These three environments might differ in the available resource to a consumer. And then if you have some consumer, maybe that consumer specializes on an individual resource. So you have a consumer, this blue bacterium, for instance, might specialize on this blue square resource. You might have this green cocci, and it might specialize on this green triangle resource. And so you get this ecological specialization, and now if habitats differ in the resources that are present, they possibly also differ in the organisms that are there. Alternatively, our consumers can be pretty generalist. They might use all the resources. They might not really, it might not matter. So you could have a situation where you have these blue square resources, these hexagon resources, or triangle resources, but since consumers are generalists, they don't really care, and there's maybe other processes deciding who's in one habitat versus another habitat. In this instance, we could also consider these resources as substitutable. To a generalist consumer, these resources are substitutable. They don't need to distinguish one or the other. And resource diversity might not infer community diversity or differences in communities between habitats. So I want to start by thinking about an important resource, maybe globally, and that has different forms. So the resource I want to talk about to start here is phosphorus. And this is a classic experiment done by Schindler in the experimental lakes region of Canada, where they were really trying to get at what causes eutrophication. And the take-home point of a lot of this is that most inland water bodies are limited by phosphorus. And if I add carbon and nitrogen here, carbon, nitrogen, phosphorus here, that phosphorus might cause a bloom. You have all these organisms that are just like taking off because now they're not limited by concentration of phosphorus in their habitat. Phosphorus is an incredibly important resource across inland water bodies, and like the ones I study, the bacteria that live in. However, the type of phosphorus usually used to study these effects, or that I might add to culture media in the lab, is usually phosphate, the most biologically available form of phosphorus, I would argue. But if I go to a lake today that has some amount of phosphorus in there, I try to find phosphate, soluble reactive phosphate, I'll find almost none. It's almost not there. Instead, there's a diversity of other phosphorus compounds. And so are these all substitutable? Does it matter what the type of phosphorus is? Is it just I need some phosphorus, or are there organisms that specialize on different types of phosphorus? So we set out to sort of explore this. We used an experimental mesocosm approach. We had these 300-liter tanks, all started off with essentially the same community, and we added a diverse mixture of microbes and phytoplankton and zooplankton. And then using a factorial design, we added different types of phosphorus. We have some replicates that have phosphate added. Pretty basic. We've had the ones where we added ATP, an organic phosphorus source, but also pretty common in biological systems. I'm sure everyone that's opened an intro bio book has heard about ATP. It's important. But you also have chemicals like phosphonates, chemicals that have a carbon to phosphorus bond, something that's really hard to break up. These are structural chemicals that plants use to sort of store phosphorus, and they're really difficult to break, and there's specialized enzymes needed to break carbon-phosphorus bonds. So these might fundamentally be different. You also have other complex molecules like phytic acid, where you have these phosphate groups, but they're sort of interacting with this phenolic ring structure that can stabilize it and make it hard to break apart. So you have these common molecules like phosphate, you have other biological molecules like ATP, and then you have these structural compounds, mostly made by plants, in order to sort of hide phosphorus away. Are these all the same, or are they different? Are they substitutable? So we formed some hypotheses to look at if these chemicals are substitutable or not. We predicted that there's maybe 3 hypotheses that we can work with. One where phosphate's different than the rest. It's inorganic, it's easy to move across the membrane, everyone likes it, that's what we use. Maybe these other chemicals are just hard to get at. So the other organic phosphorus sources might be substitutable to the exclusion of phosphate. Alternatively, maybe these biological molecules, phosphate and ATP, a chemical that all cells use and can easily be transported across the membrane like other nucleosides, like amino acids and proteins, or like other nucleosides like nucleotides like DNA. But these other structural compounds that are more plant-based are sort of hard. Like you need specialized enzymes, they might be hard to get across the membrane, they're sort of big and bulky. They might be substitutable, and the biological ones might be substitutable. And then alternatively, these molecules represent a gradient in complexity. And here I'm talking about molecular complexity. I used a measure of chemical complexity called Bert's complexity to sort of score these from the least to the most complex. And this is given the types of elements there, the type of bonds that are there, and the number of bonds, the oxidation state, things like that. There's other forms of complexity, or you can think about things like delta G, something fundamental about the chemistry of the molecule. And it can make all chemicals sort of different. So you specialize on different chemicals given their complexity. So we went up to test these hypotheses in our mesocosms, and we wanted to address 3 questions. We want to know if phosphorus resource heterogeneity alters the composition of microbial communities. That's pretty fundamental to our predictions about how resources influence communities. But then we also want to know if this goes further, if there's evidence that this heterogeneity influences microbial-mediated ecosystem processes. And I'll get to this in a little bit, but this is sort of what we're ultimately interested in if we're trying to understand why it's important to understand how chemicals might change microbial communities. So I'm going to start with the first part. And I told you that I'm going to sequence a bunch of communities, and I'm going to show you the data. I'm not going to show you the details of how communities are sequenced, but I do want to show you how I'm going to present the data. I do a lot of multivariate analyses where I'm analyzing complex data sets where I know the relative abundance of all the bacteria in all these habitats. So I have, let's say, 24 experimental units, and in them I have thousands of bacterial species and their relative abundances. And I really want to know which communities are more similar to the other, which communities are more similar. That will determine if they're using similar resources, and maybe these resources are substitutable. So what I've done is I've used measures of similarity, a measure of percent similarity called Gray-Curtis index, and its inverse, the dissimilarity. And I'm looking at the dissimilarity between all these experimental units, and I'm going to sort of reduce all of those variation into two axes. I'm going to reduce the complexity of the data that represents the differences between communities into this biplot, where you have two axes, and these capture some of the variation between communities. Sites that are close together in this space will be more similar to each other, and ones that are further apart will be dissimilar. Okay? Everyone follow that? I'm going to show you this plot a lot, but I'll reintroduce it a few times. Okay, and now what I've done is I've plotted each of the communities in this multivariate space, and I've surrounded the treatments using these ellipses. And we can ask the question, are communities different? Are the communities based on the treatments presented different? I think it's pretty easy to answer, yes. And I can use fancy multivariate statistical techniques, in particular PermaNova, permutation-based multivariate analysis of variance, to show that 34% of the difference, 34% of the variation in community composition can be explained by the different treatments of phosphorus. So we had different types of phosphorus and different microbial communities assembled. But we want to know more about that. We really want to know, are these resources substitutable? So you have maybe the treatments that phosphate, and you have a set community that assembles in them, and you have the communities with ATP, and you have a set community set by them, but are those similar? So what we did is we used that same multivariate technique to test each of our substitutability hypotheses. What we find is that there's the most support for our biomolecules substitutability hypothesis. That microbial communities in mesocosms with phosphate and ATP are more similar to each other, and microbial communities in mesocosms with phytic acid and this phosphonate resource are more similar to each other, but these are the exclusion of each other. So we have this sort of biological substitutability of phosphorus resources. And sort of the mix is sort of right in the middle. And this is pretty cool. So phosphorus resources are substitutable, but there's differences. You get different communities with different types of phosphorus. And that leads us to our next part, where the answer was yes, there's differences in community composition. And here's what I just told you without knowing I had a slide here to tell you that. There's the most support for our biomolecules hypothesis, that these biologically related phosphorus sources might be more similar to structurally formed storage molecules. But this is what I want to get at, is these ecosystem processes. All right, so here we have these changes in the microbial community. Now, does this change anything at the ecosystem level that we might be interested in? There's two things I want to talk about today. I want to talk about bacterial production. This is the amount of biomass bacteria are making per liter per hour. So how much new microbial biomass are we getting? This is that movement of resources into the aquatic food web. The other one here is growth efficiency, bacterial growth efficiency. This is how efficient I am at making that new biomass. To make, as a heterotrophic organism, if I need to make new biomass, I need energy. So I need to use some resources that are available to me to get energy. And that takes away from what I can make biomass with. I also might be wasteful and not use all my resources. But these are sort of two fundamental ways we can understand how microbes are interacting at the ecosystem level and how resources like carbon are being transformed. This is really important for global carbon cycling and carbon budgets. So when we look at these two, first thing we see is both of them increase when we add phosphorus. We knew phosphorus was a limited resource in these systems, so this was expected. If we add phosphorus, we should see increases in ecosystem processes such as production and efficiency. So we saw across all of the treatments that bacterial production rates went up. We also saw that across all the treatments, bacterial growth efficiency increased. So all the organisms are more efficiently making new biomass. But we can also ask if this follows our substitutability predictions. If similar communities are forming under different resources, do they still do the same functions the same way? If different communities are forming under different resources, are they doing things differently? So for bacterial production, we see that phosphate actually is a little higher in production rates and maybe ATP, but not really too bad. Mostly just phosphorus is higher than the other ones. But that's sort of key here, that phosphorus leads to higher rates of production than these other things. For growth efficiency, there's more of a gradient approach, and there sort of doesn't fit any of our substitutability models. So is there evidence that phosphorus resource heterogeneity alters ecosystem processes? Yes, we saw evidence that both production rates and growth efficiency were elevated when we added phosphorus to our systems. And we saw that the type of phosphorus might matter a little bit, especially for production, the amount of new biomass we're making, that biomolecules model saying that phosphate and ATP might be similar. We saw that they formed similar communities, and they also had similar increases in production rates. Efficiency was a little bit more complicated. There was maybe a little bit more of a gradient approach in there. We didn't set out to test a gradient or some kind of linear model there. We had distinct treatments, but it still increases along all of them. So take home from this part of my talk. Oh, I'm not on my screen. My keyboards must be upset. Different types of phosphorus added to ecosystems can cause drastic changes in the composition of bacteria. I didn't tell you, but there's also changes in phytoplankton communities in this system. And these changes in composition seem to be related to the type of phosphorus source we're giving them. Types of phosphorus that are more related to biological molecules yield similar communities, and types of phosphorus that are more related to structural molecules yield similar communities. And this is probably related to the enzymes needed to degrade those chemicals and access that phosphorus. Things like phosphate and ATP might be freely moved across the membrane, where these other molecules might need to be broken down outside the membrane or immediately broken down once transported across the membrane for organisms to access these phosphorus molecules. In addition to these changes in communities, we saw that production and efficiency both increased, and that at least for production, this might matter on the type of phosphorus source we're using. So in these systems where we're studying, we're interested in phosphorus inputs to aquatic ecosystems, the type of phosphorus might actually matter. And if we make all of our assumptions and all of our predictions, all of our outlook models based on the response of phosphate, we might actually be over-predicting the response of ecosystems to phosphorus additions. Okay, so that's how heterogeneity between habitats affects communities and ecosystem functions. But I told you earlier that resources are really diverse. And they're not just really diverse between habitats, but within a habitat, resources are really diverse. And you can have differences in the relative abundance of different resources, and you can have habitats that don't have one resource that their neighbors might have. Now this creates a different story. Now we can have interactions within communities, we can have different things going on, but we can still have these same fundamental processes happening. We can still have specialists. So you might have the same specialists we talked about earlier. You have a specialist on the blue square and the green triangle and the hexagon. But when one of those resources is missing, maybe the specialist on that resource isn't in that community. When one is really rare, maybe that organism that uses that resource is also really rare. So you can have interactions between the resources available within a site and the organisms there. And there might be interactions to a point where the relative abundances, the relative availability of different resources affects the relative abundances and processes of the consumers within that habitat. Alternatively, you can also have generalists, and they could consume multiple resources, just like we saw that phosphate and ATP were substitutable. Maybe a lot of these other resources are substitutable, and variation there or within the resource pool within a given habitat might not matter. The organisms there, if they're all generalists, maybe some other process controls who actually is there, or maybe they're all moving in, they just access the most available resource to them, and when that resource is depleted, they access the next one. So this variation within a habitat for resources might not matter as much. So I first want to show one approach we use to get at this. I showed you this image earlier of a gradient of carbon. Most of the dissolved organic carbon in the ecosystem is actually large, complex organic molecules that moves in from the terrestrial habitat, as opposed to more labile carbon molecules that might leak out of phytoplankton that are fixing it in the water column. So you go from this labile stuff, which isn't very chromophoric, doesn't absorb a lot of light, and it's very clear, and more of this terrestrial carbon in the ecosystem sort of darkens the water, and you can see it as this color gradient. Most of the carbon in a lot of inland water bodies is represented by this terrestrial organic complex carbon. And so we can just change the supply rates of these. You have some habitats, and they might have some amount of carbon that's fixed naturally by phytoplankton, but we could go in there and we could add more and more of this terrestrial carbon and sort of change the relative abundance of these two resources. And now what we've done is we've gone from maybe very low terrestrial carbon to a lot of it. And we've created this gradient, this diversity of different resources within habitats. So we took a bit different approach here. These are pretty large experimental units. This is a car. This is not a 96 well plate. It's a little bit larger. And we added this stuff called superhume, which is we've used as an analog for these terrestrial carbon molecules, these humic acids, and pumped it in to change the concentration of carbon in these sites. So given our predictions that there might be some kind of organisms that specialize in different types of carbon, we could predict that by changing this, we change the communities, right? If all the consumers, all these microbial heterotrophic bacteria are generalists, they might not care. They might just go from one carbon to the next. You might not see major changes in composition. But we start by seeing changes in diversity. This is sort of interesting. Here I'm showing you this gradient, the supply rate, where we added more and more carbon. This is like this gradient here. And as we add more and more, we see that the richness of the bacteria that are active, the organisms that are breaking down and metabolizing carbon, decreases. Likewise, the evenness of those communities decrease. You end up with organisms that are more dominant than others. So there's something going on here. You add more and more, if you change the resources, you go from not a lot of terrestrial carbon to a lot of terrestrial carbon, you sort of change the diversity of the bacteria. But does that sort of change the abundances of individual bacteria? Sort of in this species sorting framework, where changing the available resources might change the interactions between species, and you might get differences in who's abundant in different habitats, given the available resources. And in fact, we see exactly this. We see that along these gradients where we add more carbon, that you have taxa that become more abundant, that might specialize on that resource. And you also have taxa that become less abundant. They might not like that resource. You've sort of overpowered the resource they like, and they become less and less abundant. So there's sort of this framework that there might be bacteria that specialize on different types of organic matter. But what about the composition of this dissolved organic matter? I told you earlier that we have these great techniques where we can go in and we can use high resolution mass spectrometry, and we can identify individual molecules. And instead of just adding terrestrial carbon, which is a mixture of different molecules, we can sort of explore the diversity of each carbon molecule and see if that relates to the diversity of the bacteria in habitats. So this has sort of not been explored too much. We don't know in many habitats what's the diversity of organic matter. If you go within a watershed, do lakes in the same watershed have similar carbon molecules in them, or do they have very different carbon molecules in them? In addition, relationships between dissolved organic matter and communities is not really well resolved. We have this idea that there might be specialists, but does this really scale up to the entire community? Do you get large changes in community composition due to individual molecules being changed across habitats, even in the same watershed? So we sort of interested in these questions, we wanted to address them. This is a little bit more difficult to get at than adding a bunch of humic acids to some experimental ponds. So I took a little bit of an observational approach to this. These are the field sites Jay was talking about earlier. This is a map of Michigan. They're way up here on Lake Superior. There's 10 water bodies in this Huron Mountain region that I've sampled. And these are all connected in the same watershed, which is sort of important. There's the same dominant tree species in the forests around each of these. but there might be variation in the vegetation around them, which might be important for determining the type of organic matter in each of them. The other interesting part about this is a near pristine environment. There's never been motorboats on any of these lakes. To get up there is actually really difficult, and you don't go more than 5 miles an hour down these little dirt roads, and you don't go fishing. Like, none of this happens at these sites. So near pristine, there's not a lot of anthropogenic inputs. So we're sort of looking at natural systems, sort of natural resources that are available to bacteria, and are there connections between the diversity of those resources and the composition of the bacterial communities. Another key thing here is that there's a gradient in resource availability when we group things at the large scale, like dissolved organic carbon, total nitrogen, total phosphorus. There's a gradient of carbon from about 5 milligrams per liter to 30, and nitrogen from about 1/4 gram to 2. And the concentration gradients of carbon, nitrogen, phosphorus are all highly correlated, which is nice because we can sort of compress them down into one axis that represents the resource gradient, the concentration of resources between these habitats. And now our habitats, our lakes, represent a gradient in the availability of resources, independent maybe from the composition of the resources there. So let's sort of ask that first part question. How diverse is the organic matter in 10 lakes in the same watershed? So we use solid phase extraction to extract all the organic matter in a liter of water. We then sent it off and had high resolution liquid chromatography tandem mass spec break apart these chemicals and sort of identify them. This isn't the highest resolution we can get, but this is the highest resolution while maintaining some structural information about molecules. We can sort of look at the diversity of organic matter and also know something about the structure of that molecule. We can then use possibly databases to identify the individual compounds, and if there's important ones, we might be able to identify important compounds that that alter entire communities. And from this, we get a profile. Just like I might have a profile of the bacteria in a given lake, I now have a profile of the carbon, the dissolved organic matter resources available to those bacteria. Okay, now I'm going to go back to the community ecology thing, and I'm going to use another one's ordination techniques. This is the same ordination technique. I failed to tell you it's called principal coordinates analysis, where I'm looking at the similarities in the composition of organic matter between lakes, just like I did earlier by looking at the similarities in bacteria in those mesocosms. I have each lake plotted up here as a point and labeled, and then I have these vectors. These vectors represent individual carbon molecules in our data set. Across all the lakes, we were able to identify over 700 molecules that differed in, or that were present in all these lakes. Of those 700 molecules, about 170 are important for distinguishing lakes. And based on all the molecules actually, lakes are 30% dissimilar in the composition of organic matter. Now these are lakes in the same watershed. Some of them are connected. For instance, Upper Pine and Second Pine, these are connected. Like you row from one to the next, directly connected, but they're very different from each other in the terms of the organic matter present. All right, so we have a situation. We know we have lakes that differ in the concentration of available resources, but they also differ in the composition of those resources. Now, does the composition of these resources affect the communities? So I want to start earlier, just like I did earlier, I want to look at diversity. Sort of the same measures of diversity, richness and evenness. I want to know how many species are there and what's the relative variation in their abundances. And I want to look at this in terms of both the concentration of those resources, which I compress down into one axis, a resource concentration axis, and the richness, just the number of independent carbon molecules present. And we might ask if just the concentration or the number of resources might affect these diversity measures. If we're looking at the concentration of resources, we don't really see any changes. We get more and more and more. These are really messy figures, as Ken and Spencer do this. They're really messy. There's lots of variation, but they're not significant. There's actually like this weird situation here where these two smaller water bodies have a lot more species, but all the other ones sort of are down here and show a negative trend. In a few of these analyses, I've removed these two lakes just because the relationships are not significant either way, but they're sort of outliers in a few of these cases. All right. So the concentration of resources in these 10 water bodies, which are in the same watershed, doesn't really affect the diversity of organisms there. How about the richness, the number of different carbon molecules that are present? If we look at the richness of bacteria, it doesn't really change. Across all of our lakes, doesn't matter if we're talking concentration or the richness of organic matter, there's not a big change in the number of species present in each lake. But even this is a different story. So this is this variation in abundance. Do you have some sites where some individuals are really abundant, others are really rare, very uneven site? Or do you have a situation where things are more or less at closer relative abundances? What we see, which is really interesting, is as sites that have more DOM molecules present have more even communities. They sort of have some point in the abundance of different organisms where those different molecules might be benefiting other organisms. You have this change where communities become more even. You have less variation in the abundance of different taxa across your system. But bringing this up later, there's not a change in richness. So this is sort of going on in the middle of these communities. Now we might ask the next question. It's like, all right, we saw there's changes in diversity. In particular, there's changes in evenness, which is sort of an underappreciated measure of diversity. How about the composition? How about the actual makeup of different bacteria between these lakes? So I'm going to test two models. I'm going to plot my lakes, the bacterial communities, in this ordination space, just like I've done before. So points in space are going to represent the bacterial community. And then I'm going to use two multivariate models using a multivariate technique called distance space redundancy analysis, which just allows me to then test if these predictors, the concentration of organic matter or the composition of organic matter, and I'm going to use this sort of multivariate approximation of the composition of organic matter to simplify that. And I'm going to test to see if I can explain variation in the community composition given either the concentration of resources or the composition of resources. I apply these as vectors. They're independent vectors. The first one we're looking at is the concentration model, which is this vector here. Resource concentration overall explains 28% of the variation between communities. Pretty cool. So just knowing the variation, difference in the concentration of resources, we might be able to differentiate different communities. And you can see that these lakes over here do kind of separate out. They do have the most resources. However, composition, our second model, and I'm plotting the two axes because I use two multivariate axes to represent composition, explains 44% of the variation in communities. So now you have lakes all in the same watershed, connected, many of them are connected. They have different communities. They also have different organic matter present in those lakes. And that organic matter might explain up to 44% of the variation in communities between those lakes. That's pretty cool. Just like before, though, we can find species-specific changes in abundance. So here, what I mean by species-specific, I didn't have a treatment. What I have is maybe the abundance of a species. What's the percent abundance of taxa 1 in all the lakes? And how does that relate to the abundance of a resource, an individual carbon molecule? And here I've presented this as parts per thousand. And we can see, just like before, when we change the concentration of carbon, that you have taxa that decrease and taxa that increase alongside independent molecules. Now, I have thousands of species and hundreds of molecules, so this gets a little complicated. I don't want to show you a bunch of these plots. But I want to use co-occurrence analysis, which is just a way to look at these relative changes across all the members of the community to sort of see what's going on with all the bacteria, see if there's any patterns. So what I've done is for each interaction, so each bacteria and each independent organic matter compound, I've tested for correlations across all my habitats. I've gone through them and I've gotten rid of any that are just statistical phenomenon using some randomization techniques. So I've sort of gotten rid of anything that might just be noise, and I'm going to just look at the ones that are significant. And I'm going to have both significant positive, which could be the situation where bacteria produces a chemical, or somehow there's some facilitation between a resource and an organism. There can be negative interactions. Classic example of negative interaction is I eat my food source, it goes down in abundance. So I'm going to do this across all of my interactions. And we see a mix. We see some organisms that interact with a lot of resources and might be more generalist. We see other organisms. I have taxa on this axis and I have resources on this axis that interact with just a few molecules. They have only significant interactions with a few different molecules. So these might be very specialized taxa. In addition, of course, there's some resources that are more available to all consumers, and there's some that are pretty restricted. There might be that complex matter that's hard to break down. Now, this is a lot of data, but what I really wanted to get at earlier, what I talked to you about earlier, and I've sort of pulled through this entire talk, is generalists and specialists. Like, what is the relative importance of generalists and specialists in these communities? I find a lot of generalists that use a lot of compounds. I find a lot of specialists. But in addition to knowing these interactions, I also know the relative abundance of each of these organisms in each lake. So what I can do now is I can, given this information, and assuming maybe a cutoff to start with, I can ask, are communities dominated by generalists or specialists? If they're dominated by specialists, you see a lot of these connections between the composition of resources, here organic matter, and the composition of the consumers. If they're dominated by generalists, there might be other mechanisms explaining the abundance of consumers, but it's not really because they're specializing on different resources. So I went to each lake and I classified each tax as a generalist or specialist using a cutoff. I said, to start with, if you use 1 to 2 resources, you might be a specialist. You specialize on a few resources. If you use more than that, you might be pretty much a generalist. You can use different carbon resources. That can be really important. And I went across all of them, and what I find is that most of my communities are dominated by generalists. Taxa have interactions with a lot of different resources. Now you're a little confused because I told you earlier that composition can explain 44% of the variation in communities. So if they're all dominated by generalists, like how does that happen? Well, one explanation might be that this is pretty close to 50%, so there's still a lot of other variation, a lot of other taxa in these communities. But I want to get at something a little different. Do we know that there's differences in community composition due to heterogeneity in dissolved organic matter resources? Yes. Lakes with high amounts of DOM or high richness of DOM resources have more species evenness, which I think is really interesting, and I think we need to explore a little bit more. However, across all lakes, composition explains 44% of the variation in community composition. So there's some, at some level, resource composition matters. But these communities appear to be dominated by generalists. So this is sort of hard to pull together here. And I think that makes us come back to these results. We don't see change in richness. We can add more and more DOM molecules here, which is observational, but that might play out if we did this experimentally. We can add more and more. We're not seeing new taxa. We're not seeing taxa become rare, sort of starting to invade these communities. What we are seeing is that communities are becoming more even. So taxa that are there, when different resources are more available, they can have higher abundances. These aren't the most dominant taxa. These are sort of in the middle. These are very small changes. This is not a huge increase in evenness. So this represents sort of this middle abundance of taxa. We know that the composition of organic matter matters, and the composition of organic matter might explains 44% of the variation in communities. It might not, however, explain the concentration or the, it might, however, only be able to explain 44% of the variation because communities are dominated by generalist taxa that can sort of exist in different sites. You have half the community can just be generalists. They can move between, they can switch resources, they can use lots of different resources. So what I'd argue is that the effect of organic matter on the diversity of organisms appears to be in these middle ranks of abundance. And I'm going to borrow something from politics right now, and I'm going to call this the microbial middle class. Everyone has the rare biosphere, but I'm going to talk about these middle abundant organisms. And I would say that the diversity of here dissolved organic matter resources is really important for explaining the variation in the composition of those middle rank members of the microbial community. What we know is that there's a lot of very rare taxa. There's also a few very dominant taxa. Other work has shown, for example, work in Jay's lab, is that these most dominant taxa are not always active. They're not always the most active members in the community, they might not be doing all the biogeochemical cycles. So to argue here is that this is proof that it's these middle abundant organisms that might be more responsible for the biogeochemical functions happening between habitats. Resources might have a larger impact on their abundances, and we might need to actually look more, not the most dominant organisms or the most rare organisms. We need to focus more of our attention on these sort of middle abundance organisms in order to understand how changes in communities and variation in communities between habitats affects ecosystem functions and biogeochemical cycles. Okay, that was a lot. Take home. Dissolved organic matter is highly diverse. Even within the same watershed, you can have 37% variation in dissolved organic matter composition. Variation in the dissolved organic matter alters the community composition. At least we've shown this in experimental manipulation of one resource or a mixture of resources, and we've shown it as correlations between the abundances of different taxa and different resources. We've also shown that sort of generalists might dominate and specialists might comprise sort of the middle ranks of these communities, which I'm going to call the microbial middle class for now. So as I wrap up here, I think I've convinced you that resources affect microbial communities. And this might not be like, you might be sitting there as a community ecologist, be like, yeah, I sort of would have predicted that. But I think at this level, understanding this diversity of resources and how microdiversity of resources and how different forms, chemical forms of things like phosphorus and carbon affects communities is sort of instrumental in how we understand how microbial communities and ecosystem functions might change in the future. I've shown that there's heterogeneity between habitats that matters, and there's also heterogeneity within habitats that matters. I think this is both key as we move forward to explore communities across the globe. What I know after putting together all my dissertation research is that there's really complex interactions between microbes and molecules. Like it's so complex, you might argue that it's not even worth getting into. One thing I can tell you right now is there's no glucose, sucrose, acetate, butyrate. None of these resources that we use in the lab, they're not present in the environment. None of our mass spec could detect them. In fact, the resources we were detecting in the mass spec, we used 3 different databases to try to identify them. We couldn't identify 99% of them. No idea what they are. 700 molecules in lakes, I have no idea what they are. No one knows what they are, but someone's using them. Some bacteria is breaking that down somehow. Extracellular enzymes, specialized transporters and degradation enzymes within cells. We haven't explored any of this variation in microbial communities. We don't know how these microbes function or what sort of technological advances we might have by sort of understanding their physiology. We know there's resource heterogeneity within and between habitats, and this alters communities and alters the processes these communities perform. Specifically, one of the things that I've looked at a lot is the efficiency of these organisms to grow, how efficiently they make biomass. I showed you earlier that things like phosphorus resources might affect how efficiently microbes grow, but I don't have time to get into this, as you know, but individual resources might affect how bacteria grow and how efficiently they can grow. So this diversity of organic matter is really important for structuring communities, but it's also important for their functions, and I'll be happy to talk to you guys more about that later. And with that, I would like to entertain any questions. Thank you. Yes.
So within a lake, what point is the minimum distance where you can say that this dissolved organic matter doesn't change? Like what's a micro environment where it might be constant?
You're asking if the organic matter is constant?
No, I'm saying what about like the minimum distance where you can say with some confidence that there won't be a change in composition?
Oh, I'm not sure. I think this begs some experimental manipulations of resources.
Yeah.
And some people have gotten at this. Some people have done transplants where they've taken the microbes from 1 liter of water from like one lake and put it in a liter of water from another lake and seen how communities have changed. But I think this is really a ripe area for people to do more research. I think people are, and these are really hot topics. And the reason this is important is because we're changing the globe, naturally, anthropogenically, what have you. The communities of plants are moving. You have tree species that are sort of replacing other tree species, and as this happens, there's new chemicals that go into these water bodies. So when do they change? At what point do changes in plant communities affect the DOM so much that the communities change differently? We showed in that study where we added more and more terrestrial carbon that we stabilized communities. We made it so the bacteria got to a point where we could add resources like phosphorus and they didn't do anything. They didn't change in abundance. This is really bad because then the phosphorus hangs out and you have all sorts of eutrophication, other things that happen. So these changes in resources can have big changes on the function of the microbes, but we don't know at what point, when you get them, what types of resources you need to add, and how substitutable different dissolved organic molecules are. So I wish I could answer that, but I can't right now. I think James had his hand up first.
I have two questions.
Yeah.
One of the methods questions. I'm wondering about the sampling of your system. You can go in and get a thimbleful of water and look at bacteria in there and the DOM in there, or do you filter liter after liter after liter and concentrate it? And it seems like that would change your result. So then my second one, before you touched on the idea of algae, but you didn't really talk about the diversity of algae, and I wondered about the possibility that algae might be mediating some of these changes, that it's not going to be the bacteria, that you throw in the resource and it changes the algal component, and then that changes the bacterial component. Like when you define the containers, you could somehow remove the algae regularly to somehow inhibit them and see if those changes in growth rate from early on they would be the same.
Yeah, so the first one, I love my job because I get on like Canoe and I go out to the water and I use sampling devices that collect a couple liters of water at once. And when I did those studies where I looked at the diversity of organic matter, I used the same water sample. I took 2 liters of water, I put 1 liter in this bottle and 1 liter in this bottle. They were all sort of mixed, so they're somewhat homogeneous. And 1 liter I used to sequence the community, and the other liter I extracted all the organic matter from it to look at the DOM diversity. And there might be micro variation even within the same water body of that organic carbon. I tried to sample all these water bodies in a deeper section, stay away from the shore where things might be sort of weird. You might have a lot of litter, you might have a lot of detritus floating. But I did have to, in the classification of organic matter, filter out large debris. So I filtered it through a 1 micron filter to get out any large particles. So I'm looking at just the dissolved stuff. And then it's captured on solid phase extraction, and it's what extracts in methanol. So large particles could get stuck in the filters as well. So I think that addresses the first question, more or less. The second question was about the phytoplankton. So in the second study where I looked between the lakes, the concentration of phytoplankton, we approximated by measuring chlorophyll. And it's highly correlated also with carbon, nitrogen, phosphorus. So we explored that a little bit, but essentially any inputs that algae might have, and we're assuming all algae are the same here, which is a gross assumption, but might be captured along that resource gradient and not the compositional changes. At the same point, I would predict that a lot of this variation in organic matter is due to phytoplankton. There's no way to sort of parse out using these techniques, this is what comes in from plants, this is what's fixed in the lake. But I think it's important to look at the diversity of all this organic matter together. Maybe there's ways in the future where we might be able to parse apart what's due to algae and what's not. Maybe with experiments where you can eliminate the algae and see how the resources change. And let's say a bag experiment where you're looking in a water body, you might be able to exclude the algae by shaking them out or something. In the experimental approach, we analyze the algae as well. In fact, we use the chloroclast sequences that we capture when we do 16S to identify each of the algae and look at changes in the community, in the algal community, as well as changes in the bacterial community. We saw similar changes according to the biomolecule substitutability model, I believe. I have to double check on that. But we did see changes in the phytoplankton communities in addition to the bacterial communities. And there we were really interested in changes throughout the whole food web. In fact, in that paper, if I can go into this more, but we looked at from bacteria to zooplankton and how the whole food web changed when we manipulated these phosphorus resources. So we weren't really interested at the time in just pinpointing only the bacteria. We really want to know how everyone changed. But you could shade the mesocosms and exclude the phytoplankton. I'm going to go to Megan, who's...
Okay, so one of the things is when you go and you sample a lake, right, the resources that you detect there, the idea that you don't see anything like resource, right, it's either because it's not important at all and it's just not in a lake, or that they're very important in these organisms that are particularly carbon limited. And so now you have these 700 like, molecules that you see that are in the lake, that it could possibly be that these aren't affecting the community at all. They just happen to be characteristics of the lake because the environment that lake is in is runoff and things that are in this lake. So how much do you think maybe like bacterial behavior of like communities that are already established in there, maybe like more aggressive bacteria that are clearing their area, things like, collagens and things like this, that they're able to compete better, and that's going to affect the microbial community and like the proportions of microbials in these lakes, microbes in these lakes, as opposed to the actual resource.
Yeah, so I'm going to sort of take that in a little bit of direction. I'm going to assume that there's some bacteria that love glucose. We can just consume as much glucose as we want. We might even ferment that glucose. We want that glucose so bad, we want to exclude our neighbors from that glucose, that we might even use it inefficiently in order to increase our abundances. We might be the dominant organisms in those habitats. What I'm saying here is that the variation in these intermediate classes, these might be the organisms that aren't really good at competing for those resources, and now they're left to try to specialize on what's left. Because there's a lot left. There's 25 milligrams of carbon per liter in these high abundance lakes. There's no glucose. So what are you going to do? You're going to wait for some glucose to come around, or are you going to try to use those other molecules? We know that there's extracellular enzymes that break down complex organic matter. We know that there's bacteria that can specialize on even man-made complex organic matter and degrade things. We find these all the time, so organisms that break down aliphatic acids. And I think what's going on here is that the organisms that are going to sort of rely on those labile compounds and just eat them up as fast as they enter might be those dominant organisms. That might be absolutely true. This sort of follows competition experiments, where you might say like somebody who responds quickly and consumes all the resource, drives the concentration down to exclude its neighbors. They might have high peaks in abundance. I can't look at that here because I couldn't isolate those resources because there's not any, but I do understand that is a mechanism that can be happening in these communities.
Do you think like a bigger driver would be the fact that you have these middle-class individuals that are able to invade because they are more generous, able to break down these things, or do you think it's regulated by these upper-class microbes who are now forcing these guys to find a secondary resource?
I think this is just how ecological specialization has played out, that organisms have evolved mechanisms to use different types of resources. They might still be able to use glucose, but in these systems, they might just prefer, or it might be more beneficial for them to hang out and use these other resources. Now there is some positive interactions. There's probably a lot of microbially produced chemicals here. We use some, I know you saw these graphs where we use some network analysis to look at interactions where you have this bacteria has a negative interaction on this resource, but that resource has a positive interaction on this bacteria, and you can have these sort of interactions. We sort of think of all microbes as like one trophic level, but instead there's interactions between molecules. You have synergies. You have bacteria produce a chemical that might be used by another bacteria. I've tried to explore this a lot. I'm happy to keep trying to explore it. Come back. But yeah, it's very complicated. But I think the point is that the diversity of organic matter, the standing diversity of organic matter might influence more of these intermediate abundant organisms. Anyone else? I love this stuff.
You're gonna get some.
I know, I'm afraid that Spencer's got a grumpy face on. Okay, well, thank you. Thanks for the feedback. Bye. Hey, by the way, there's 11 people watching this.